Hey everyone. For those of you who don't know me, I'm Jennifer Sloan and I work at the NIH with Dr. Venditti and Dr. Manoli, and I've been seeing patients with MMA and cobalamin disorders for almost 18 years now. But today I'm actually going to use my researcher hat and talk more about what we do in the lab. And so my task is to talk about mouse models for our group of diseases. And thanks to Brittany for this slide about the research map. And I'm just showing you what I'm focusing on today, which is, what mouse models are available for cobalamin disorders and MTHFR, and like what are some new therapies. So I'm going to just try to explain a little bit about why we might need animal models. I know Dr. Chapman said obviously they're not humans, but they are useful for us studying new treatments and the pathophysiology of our diseases. And then I'll also, after that, tell you a little bit about our mouse model for cobalamin C and some efforts that we have on gene therapy. So why do we need animal models? And as Brittany talked about, there's so many unanswered questions about our diseases. Like what happens in the tissues and cells, like in cobalamin C, what cells are affected in the eye, for example? What's happening to the brain? We don't have answers to these questions, and so animal models can help us understand the disease pathology. What is causing the symptoms? Is it just the elevated MMA or homocysteine, deficient methionine? Is it methylation? We don't understand hardly any of the disease mechanisms, and this is where cell models and animal models can really, really help us to dissect out these things and lead us to new therapies. And then, obviously, to test current and future therapies. As Dr. Manoli showed you with the B12 dosing, we don't really know the exact dosing. We don't know the dose for betaine. Does it even work? So these are some things that we could try to answer with animal models. And there are very few clinical trials, as Brittany showed very nicely. Unfortunately, we need the animal models really to do the work to move things more toward the clinic. And so there's options now. If you guys get a chance to see all the wonderful booths outside, those are companies, many of them, that have clinical trials. And so it's a really exciting time, and I think we can eventually get there and move forward with new therapies for our diseases. I'm going to talk a little bit later about AAV gene therapy. So just a couple animal models that we often use are zebrafish and mice. And so just to give you guys a little bit of background, the zebrafish are useful because we can manipulate their genome. We can make them have, cobalamin F or J, and we can get tons of embryos to study. So if we put two fish together, we can get hundreds of little baby fish to study, and we can watch their development over time, and all the organs develop in 4 days. So it's pretty quick and useful, but they're limited. We can't really give them injections, right? So we can learn something from zebrafish, but they're a little bit more limited than mice. And mice are obviously have a lot of similar organ systems to us. We can also manipulate their genomes to make a mouse with cobalamin C, for example. They're more similar to the humans in terms of anatomy and physiology, and they reproduce quite quickly. We can get, a litter of pups in 21 days, so we can make as many mice as we want. But they do have a shorter lifespan, 2 years. So there is some maintenance. And oh, sorry, I forgot I had this nice slide just showing the development of a zebrafish. And so how do we make animal models? And it's very complicated, but just briefly, you may have heard of CRISPR-Cas9. This is a new technology, which is basically like a genetic scissors. So we can say, hey, genetic scissors, go to the MMA-CHC gene and cut it and make a mutation or a variant in that gene. And that's pretty much what we do. So we can genetically engineer various animal models, either fish or mice, and that helps us a lot. And so I reviewed the literature for this talk just to see what animal models are out there for cobalamin disorders, and I made a list, which I'm not going to go through in great detail, but I can share it with Brittany, obviously, for the future. There are quite a number of animal models, and you can see them listed here for your favorite cobalamin disorder, MTHFR, and I list the references, but for some there aren't any animal models. For some there's just zebrafish. So sometimes we have a lot of limited information about animal models in cobalamin disorders. And the main problem, and here's just the rest of the pathway, including the transcription factors, cobalamin X. So the main problem with I summarized it here. Sorry, I'm having trouble. Oh, there we go. Okay. So here's the list. So we don't have all of them. And the major problem is that many of them are embryonic lethal, so they don't make it to birth. So I just showed here the development of a mouse from one, conception to 15 days. And, for the example, the F, G, and cobalamin C mice, they all don't get born. So if a mouse doesn't get born, we can't really study it. So it's a really big challenge in our field, and we don't really understand why, but obviously vitamin B12 is very important for development in mice. In humans, we know they get born, and we see patients, after birth. But in mouse models, it's just a little bit different. Again, the differences between human and mice. So that's a huge limitation. And then when we do have mouse models that are viable, so like here's an example. On the left is a mouse model of cobalamin C, and you can see the embryos are barely getting made. So no pups get born. So what we can do is overexpress the MMA-CH gene. We can make all different flavors of mice. So the purple is like we knocked out the gene completely, but then we can give back the gene for a transgenic mouse, or we can make a tissue-specific mouse. So we can make a mouse in which the gene is only knocked out in the brain or in the eye, for example. And so we can kind of give back more of the gene that's not working, but we can get other, maybe a surviving animal. But this is an example of the mouse model for cobalamin C. It actually doesn't have any of the phenotype or any of the symptoms of cobalamin C once you give it that correction. So it has the biochemical features there on the right, but the mouse is looking and developing normal. So we have some mouse models, but they're really not optimal because we can't really study much of the therapy. There's no problem with the growth or the eyes in that mouse, and so it makes it really, really challenging. And so I looked in the literature to see, what treatment studies have been done on the mouse models that are available, and there's so few. And so this is where your your research map comes in, is we need to have some more research, basic research, developing animal models and treatments. And so there's really one animal model of cobalamin G where the gene is missing from the brain, and they're able to study some of the pathophysiology of that, and they're able to come up with a medicine that improved the behavior of the mouse. And this figure is just showing the water maze, and you can see when that mouse was treated with the medicine, the SERT activator, it improved the behavior of the mouse. But this is one thing that could potentially be used potentially for cobalamin G, but it doesn't study all the other aspects of cobalamin G, right? Because the brain is only affected in this mouse. We don't know how that would affect its metabolism or any of the other symptoms. And then for MTHFR, there's a few studies, one treating with betaine and another with methyl tetrahydrofolate that does improve some of the outcomes in that mouse model. But those were really the only studies, unfortunately, that I could find for treatments in our cobalamin disorder pathway. So just to give you a little story from our lab, as many of you know, we work on gene therapy. And this is just a review article from Dr. Venditti, just showing all the different studies we've published on gene therapy for a related disorder called methylmalonic acidemia, the MUT type. And he's talking about that in the other room right now. So we have an expertise in MMA, and in addition, there's tons of gene therapy trials now. And I just listed a review article on some of the retinal gene therapy studies, because there is an FDA-approved retinal gene therapy, which is exciting, and lots of research on gene therapies for eye diseases and all other metabolic diseases as well. So this was a really exciting thing to think about for cobalamin C and cobalamin disorders in general. So just quickly to kind of briefly overview that what is AAV gene therapy. It's basically having the DNA of the gene that somebody's missing that's functional, and we package it inside a helper virus that is not causing any disease, and we inject that usually by IV. This is an example of hemophilia. And that virus goes into the cell of the liver and starts making the enzyme that the patient or the mouse is missing. So that's sort of what's going on in AAV gene therapy. So in order for us to really test AAV gene therapy for cobalamin C, we had to make a mouse. As I told you, all the other mice either are not getting born or were too mild to really study. So we made our own mouse animal model, and we use our genetic scissors, and you guys can see, some of the DNA letters are missing. I won't go into the details, but happy to talk about that anytime. So we were able to cut the MMA-CHC gene, and we were able to somehow amazingly get mice that were born, which was a huge feat for this gene and this disorder. And you guys can see on the left the pups or the baby mice with cobalamin C. You can pick them out of the litter right away because they're lighter in color. So those are the ones with the star. And then you see this little pink one. That one is very delayed. It's much smaller than its litter mate. So that one also has cobalamin C. So we were able to get mice, but unfortunately they didn't survive very well. This is without any treatment. So most of them were passed away by age 1 month. And so, we studied the ones that lived, obviously, and they were small compared to their brothers and sister mice. They had the biochem findings, so they had the elevated MMA, homocysteine, and low methionine. And when we looked at them under the microscope, they did have some of the findings that we see in the patients. In the brain, we saw hydrocephalus, which can be seen in patients that are untreated. We found some fatty liver in the mice liver. Unfortunately, we didn't find any pathology or any differences in the retina, which was surprising. But another group in Switzerland also showed the same thing when they made an animal model that was just knocked out. The MMA-CH gene was missing in the eye. They also saw the same thing, that the eye was normal. So this again highlights the challenges, like using a mouse model for a human disease. It doesn't always recapitulate all the findings that we see in the mice. But they were still useful to test one thing, what we've been wondering, and I think we know clinically, obviously, the vitamin B12 is working, it's lowering the biochemistry, but what does it do for the survival of our mice? And you can see if we treated them prenatally, so we gave the mama mice vitamin B12 injections, that prolonged their survival in the light pink. But if we gave the B12 injections every week, we could get really long-term survival over a year for the cobalamin C mice. So this kind of proves, maybe for the first time, that the B12, hydroxycobalamin, is actually working. We don't know the details of why it's working, but it certainly is improving the survival of the mice, which is wonderful. And it also improved the biochemistry. So you can see the MMA is, I don't know if you guys can see my cursor there. I guess you can't. Sorry, on the left graph, the ones in red are B12 treated, and the MMA is much, much lower than the untreated in gray. And the homocysteine doesn't change as much. I think that has to do with the thing about homocysteine metabolism in mice. So this was really exciting. For the first time, we could show the vitamin B12 really kind of helped and really treated our animal model. And we also then obviously went to try gene therapy because we've been working on that. And how we did this was we designed an AAV that has the human gene is in blue and the mouse gene is in green. And we delivered this is just a mouse pup in the middle. We delivered this by injecting it into sort of the belly, or we tried to target the liver in the mice and see what happens. So what happened to their survival? We saw the same thing, a very long-term correction of the mice. Their improved survival, they can live out to a year with a single injection into their liver. So it's really, really exciting. This is with no B12. They could live long term. And in addition, if we combine the therapies, if we gave prenatal hydroxycobalamin and AAV9 at the same time, we saw an improved survival of the pups. So it really the combination therapy seemed to work very well for the mice. So this is really exciting because it's just a proof of concept study to show that the AAV might be possible someday in the future for cobalamin C, for example. And once, all these other disorders like MMA, cobalamin C would have a AAV-related treatment that paves the way for other disorders. And in addition, we saw that the weight was improved compared to untreated. So you can see all the various treatments. The red, again, is the B12 for obvious reasons. And then the gene therapy, the weight was pretty much equivalent. And look at the mouse, how much better it looks compared to its untreated mouse, and I showed a few slides ago. So you can still see it's a little bit smaller, and it does have this interesting lighter pigmentation that we see, which we haven't quite figured out. It has lighter ears and tail. But other than that, they survive long term. And remarkably, the MMA level is quite well corrected. You can see in the B12 as they get older, these have elevated MMA. It's pretty elevated, like 1,000 micromolar, and these are down in the hundreds. So it's long-term correction of the metabolic defect. Again, the homocysteine doesn't seem to change as much. So just in summary, we developed a mouse model for this cobalamin C, which is great because for some reason that we don't understand, mouse models for other cobalamin defects and even cobalamin C itself have been really challenging because they're mostly lethal and the mice don't get born. So we developed this mouse, and we were able to show really that the B12 improves the survival and the metabolic phenotype. And comparable to that, really just a single AAV injection really provided long-term correction compared to weekly injections of vitamin B12. And so it's a really exciting proof of principle study to show that AAV gene therapy could be possible for cobalamin C and other defects in the future. And so with that, I'm happy to take any questions, just thanking obviously the HCU Network America for this opportunity to give this talk today, and OAA and all the families. Really, this is why we do this work, is for you, and you inspire us every day. So we really appreciate the opportunity, and I'm happy to take any questions.
Yeah, we're going to have to hold the
Okay.
questions till the panel.
Sounds good.
We're starting another session here
Okay.
2 minutes from now.
Great. Thank you.